Dear students, let's discuss about cell cycle and its regulation. Introduction. The cell cycle is an ordered series of macromolecular events that lead to cell division and the production of two daughter cells, each containing chromosomes identical to those of parent cells. The details of cell cycle vary from organism to organism and at different times in an organism's life. However, certain characteristics are universal. The basic fundamental function of cell cycle is passing on of its genetic information to the next generation of cells. To produce two genetically identical daughter cells, DNA in each chromosome must be faithfully replicated to produce two complete copies, and the replicated chromosomes must be accurately distributed to the daughter cells. The events in a cell cycle can be divided into four major phases. They are G1 phase, S phase, G2 phase, and M phase. The G1 phase, S phase, and G2 phase are also known as interphase, while M phase is dividing phase. In a cycling somatic cell of eukaryotes, synthesis of different types of RNAs as well as proteins, which are required for the growth of the nucleus and cytoplasm of each daughter cell towards their mesosis, are done during G1 phase. During this phase, enzymes like DNA polymerase, necessary for DNA synthesis of the next stage, and many cell cycle regulatory proteins are also synthesized. The chromosomes are duplicated during S phase. Further synthesis of RNAs and proteins that are required for the growth of the cell is done at G2 phase. The duplicated chromosomes are distributed to daughter cells during M phase. Cell cycles can range in length from as short as 30 minutes in a cleaving frog embryo, whose cell cycles lack both G1 and G2 phases, to several months in slowly growing tissues such as the mammalian liver. With a few notable exceptions, cells that have stopped dividing, whether temporarily or permanently, or in the body or in culture, are present in the stage preceding the initiation of DNA synthesis. Cells that are arrested in this state, which includes majority of cells in the body, are said to be in the GO state to distinguish them from the typical G1 phase cells that may soon enter S phase. A cell must receive a growth-promoting signal to proceed from GO into G1 phase and thus re-enter the cell cycle. The events of the cell cycle are precisely controlled to ensure the duplication of the chromosomes and their segregation to the daughter cells occurs in proper order and with extraordinarily high fidelity. The master controllers of these events are a small number of heterodimeric protein kinases that contain a regulatory subunit called cyclin and catalytic subunit called cyclin-dependent kinases. These kinases regulate the activities of multiple proteins that are involved in the cell cycle by phosphorylating them at specific regulatory sites, activating some and inhibiting others. The concentrations of the cyclins increase and decrease as cells progress through the cell cycle. The CDKs have no kinase activity unless they are associated with different cyclins, and the associated cyclins determines the proteins to be phosphorylated by a particular cyclin CDK complex. Different organisms use different cyclins and cyclin-dependent kinases. Some of the selected cyclins and the cyclin-dependent kinases are given at table 1. Now let's discuss about the regulation of entry of the S phase. In higher organisms, the regulation of cell cycle is primarily achieved by controlling of synthesis and activity of G1 cyclin CDK complexes. Extracellular growth factors act as mitogens and induce the synthesis of G1 cyclin CDK complexes. The activity of these and other cyclin CDK complexes are regulated by phosphorylation at specific inhibitory and activating sites in the catalytic subunit. Once the mitogens have acted for a sufficient period, the cell cycle continues through mitosis even when they are removed. The G1 cyclin CDK complexes prepare the cell for S phase by activating transcription factors that promote transcription of genes and coding enzymes required for DNA synthesis, and the genes encoding S-phase cyclins and CDKs. The activity of the S-phase cyclin CDK complexes is initially held in check by inhibitors. Let in G1, the G1 cyclin CDK complexes phosphorylate the S-phase inhibitors and stimulate their polyubiquitination. The polyubiquitinated S-phase inhibitors are subsequently degraded by proteasomes, releasing the active S-phase cyclin CDK complexes. Most of the current understanding of the molecular mechanism controlling entry into S-phase and the control of the DNA replication comes from the genetic study of the Saccharomyces cerevisiae. 3 types of cyclins, cyclin 3, cyclin 1, and cyclin 2, and 1 CDK CDC28 are responsible for the entry into the S phase. In wild-type cells, cyclin 3 mRNA is produced at a nearly constant level throughout the cell cycle, but its translation is regulated in response to nutrient levels. The cyclin 3 is a highly unstable protein, and its concentration fluctuates with the translation rate of cyclin 3 mRNA. Once sufficient cyclin 3 protein is synthesized from its mRNA, the cyclin 3 CDK complex phosphorylates and activates two related transcription factors called SBF and MBF. These induce transcription of cyclin 1 and cyclin 2 genes, whose encoded proteins accelerate entry into S phase and stimulate transcription of several other genes required for DNA replication, including genes encoding DNA polymerase subunits, DNA ligase, RPA subunits, and enzymes required for deoxyribonucleotide synthesis. The MBF transcription factor also stimulates transcription of CLB5 and CLB6 genes that encode S-phase cyclins, CLB5 and CLB6 proteins. These S-phase cyclins are inactivated immediately after their synthesis by binding of an inhibitor called SYK1, which is expressed in late mitosis and in early G1. Entry into S phase is achieved when the SYK1 inhibitor is phosphorylated by G1 cyclin CDK complex and subsequent ubiquitination and degradation. Once SYK1 is degraded, the S-phase cyclin CDK complexes induce DNA replication by phosphorylating multiple proteins called pre-replication complex, which is bound to replication origins. Thus, the cell enters S phase. The pre-replication complex proteins involved in the initiation of DNA replication remain phosphorylated till late G1 phase, and they cannot rebind to the origin of the replication. At late G1, these proteins are dephosphorylated by G1 CDK complex and they bind to the replication origin. Thus, DNA is replicated only once in a cell cycle. Now let's discuss about the regulation of entry into M phase. The entry into M phase can be regulated by different mechanisms. In Saccharomyces pombe and Xenopus laevis, one of the mechanisms for the regulation of entry of the cell cycle into M phase is the regulation of the concentration of the mitotic cyclins. CDC13 and cyclin B are mitotic cyclins for S. pombe and Xenopus respectively. Increase in the concentration of these cyclins along with their associated CDKs leads into the entry of the M phase, while the decrease causes the exit of the M phase of the cell cycle. The heterodimer of mitotic cyclins along with the associated CDKs are known as maturation promoting factor or mitosis promoting factor. Another mechanism for the regulation of entry of the cell cycle into the M phase is the regulation of the kinase activity of the MPF. In S. pombe, the activity of CDC2, the CDK subunit of the MPF, is regulated by phosphorylating and dephosphorylating at the specific regulatory sites by different enzymes. Phosphorylation of inhibitory tyrosines at the 15th amino acid residue in the CDK subunit of the MPF inhibits the activity of the MPF, while the removal of this inhibitory phosphate as well as phosphorylation at the activating threonine residue at the 161st amino acid of the CDK activates the activity of the MPF and thus leads the cell into M phase. One of the targets of the MPF is the nuclear lamins. Early in mitosis, MPF phosphorylates specific serine residues in all the three nuclear lamins, lamin A, lamin B, and lamin C. Phosphorylated lamin A and lamin C dimers are released into the solution. The depolymerization of these nuclear lamins leads to disintegration of the nuclear lamina meshwork and contributes to disassembly of nuclear envelopes and entry of the cell into M phase. MPF catalyzed phosphorylation also plays a role in chromosome condensation and formation of mitotic spindle apparatus. In Xenophus, a multiprotein complex called condensin is phosphorylated as cells enter mitosis. The phosphorylated condensin complexes bind to DNA at intervals along the chromosome scaffold. Self-association of the bound complexes via their coil domains and the supercoiling of the DNA segments between them are supposed to cause the condensation of the chromosomes during mitosis. Now let's come to the regulation of the progress of anaphase. The progression of cells into anaphase is initiated by unlinking of the sister chromatids. As the cells progress into metaphase, the sister chromatids are held together at their centromere and multiple portions along the chromosome arms by a protein complex called cohesins. Among the proteins composing the cohesin complexes are members of structural maintenance of chromosome proteins. The cohesin SMC proteins bind to each sister chromatid. Other subunits of cohesin, including SCC1, then link the SMC proteins, firmly associating the two chromatids. The cross-linking activity of cohesin depends on another protein called securin. Prior to anaphase, securin binds and inhibits a ubiquitous protein called separase. Separase is a protease which clips the SCC1 and helps the separation of the sister chromatids during anaphase. But by binding with securin, separase activity is inhibited till all the kinetochores of the sister chromatids have attached to the spindle microtubules. Once all the kinetochores of the sister chromatids have attached to the sister microtubules, a protein called CDC20 directs another multiprotein complex called anaphase promoting complex to polyubiquitinate securin and its further degradation. The degradation of securin relieves the separase from its inhibition and clips the SCC1 protein, breaking the protein's cross-link between the sister chromatids. Once this link is broken, the poleward force exerted on the kinetochores can move the sister chromatids towards the opposite poles. Thus, cell enters anaphase. Now let's come to regulation of the exit of mitosis. Late in anaphase, the APC also directs polyubiquitination and subsequent proteasomal degradation of the mitotic cyclins. Polyubiquitination of the mitotic cyclins by APC is inhibited until the segregating chromosomes have reached the proper location in the dividing cell. This is done by binding the APC with CDH1 protein. Once the chromosomes are segregated properly, CDH14 phosphatase enzyme removes the inhibiting phosphate group from CDH1 that enables it to bind to APC. The activated APC now directs the polyubiquitination of mitotic cyclin and its subsequent degradation. Degradation of the mitotic cyclins leads to inactivation of the protein kinase activity of the mitotic CDKs. The resulting decrease in the mitotic CDK activity permits constitutively active protein phosphatases to remove the phosphates that were added to specific proteins by the mitotic cyclin CDK complexes. As a result, the now separated chromosomes decondense, the nuclear envelope reforms around daughter cell nuclei, and the Golgi apparatus reassembles during telophase. Finally, the cytoplasm divides at cytokinesis, yielding the two daughter cells. During early G1 of the next cell cycle, phosphatases dephosphorylated the proteins that form pre-replication complexes. These proteins had been phosphorylated by S-phase cyclin CDK complexes during the previous S phase, and their phosphorylation was maintained during mitosis by mitotic cyclin CDK complexes. As a result of their dephosphorylation in G1, new pre-replication complexes are able to reassemble at replication origins in preparation for the next S phase. Phosphorylation of CDH1 by G1 cyclin CDK complexes in the late G1 inactivates it, allowing accumulation of S phase and mitotic cyclins during the ensuing cycle. Coming to the conclusion, the cell cycle is an ordered series of macromolecular events that lead to cell division and production of two daughter cells containing chromosomes identical to those of parent cells. Generally, the events of cell cycle consist of four phases: G1 phase, S phase, G2 phase, and M phase. These events are strictly controlled to ensure that they occurred in proper time and in correct order. The central components of the cell cycle control system are the cyclin-dependent kinases, which depend on the cyclin subunits for their activity. Oscillations in the activities of the various cyclin CDK complexes control various cell cycle events. Thus, activation of S-phase cyclin CDK complex initiates S phase, while activation of M-phase cyclin CDK complex triggers mitosis. The mechanisms that control the cyclin CDK complexes include phosphorylation at the activating sites, the phosphorylation, binding of CDK inhibitors, proteolysis of cyclins, changes in the transcription of genes encoding CDK regulators. Ubiquitin ligases APC and SCF also play a crucial role in the regulation of cell cycle by catalyzing ubiquitination and subsequent degradation of specific regulatory proteins that control the cell cycle. the passage of the cell cycle through G1 to S phase, metaphase to anaphase, anaphase to telophase, and cytokinesis is an irreversible process, as this involves regulated degradation of proteins. As a result, cells traverse the cell cycle in one direction only. Also, the replication of the DNA and duplication of the chromosomes is done only once in a cell cycle, as the replication at its origin is initiated only once during the cell cycle.